Hello, welcome again for the lecture of cell biology. This is lecture 9th. We are starting new chapter. The chapter 9th, it is cell cycle. Cell cycle include the stages of cell cycle, that is G1, S, G2 phases, as well as the cell division, that is the mitosis. So it also includes the mitosis, meiosis, differences between mitosis and meiosis. Myself, Dr. Sunil Patil from Loknethe Venkatrao Hire College, Panchavati, Nasik. So this is my actually video 9th for this series of lecture from the cell biology. In this video, we are going to see the cell cycle and cell division. The cell division includes two things. In detail, we are going to see the various phases of cell cycle and mitosis in this video. In the next video, we are going to see meiosis and role of centriole in the cell division. So these two things we will see in the next lecture, that is the lecture 10th in the same chapter. Today we are going to see the various phases of cell cycle and mitosis. So I think you are concentrating on this point, the cell cycle and mitosis today. It is important part, main bulk of the cell biology is the cell cycle and cell division. So you have to concentrate on each and every point from this chapter. The PDF form of notes also provided for the same chapter. All the video lectures I have also made its PDF form. So you have to take from a respective teacher and go through this. So in the cell cycle we are going to see the G1, S, G2 phases as well as the mitosis. Then we are going to see the meiosis in the same chapter and role of centriole in the cell division. So centriole role is different. So we are going to see the cell growth checkpoints, then DNA synthesis in the S phase, then mitosis with the checkpoints. So let's start. So concentrate by starting from the meaning of the cell division. So let's start the meaning of cell division.
Cell division, particularly the body of all living organism produced by growth and division of pre-existing cell. जो सेल पहले से होती है उससे ही नई सेल बनती है, either it is single cell or it is multicellular organism. Single cell zygote shows growth and development into multicellular organism by cell division. It is also called as the cleavage. All the processes including cell division, DNA replication, cell growth. Cell division, DNA replication, cell growth takes place in coordinated manner to ensure correct division and formation of progeny cells containing intact genomes. Somatic cells divide by the process of mitosis to form body cells, whereas germ cell by the process of meiosis to form the ova or spermatozoa, that is gametes. The germ cell form the gametes, male and female gametes, the female gamete and this is male gamete, and the body cell, that is somatic cell, they multiply, they divide by the process of mitosis. The amitosis, it is a kind of cell division. In amitosis, during amitosis mode of cell division, nucleus elongates and constriction appears. That constriction deepens and divide the nucleus into two daughter nuclei. Then it is followed by the division of cytoplasm and then it results into two daughter cell. It is a mitotic cell division. In amitosis occur mainly in the unicellular prokaryotic cells and the poietal membrane cells. We are starting or forwarding toward the cell cycle. अब हम cell cycle देखेंगे. Cell cycle, it is the sequence of events by which a cell duplicates its genome, synthesizes the other constituents of the cell and eventually divides into two daughter cells. It is termed as cell cycle. Sequence of events by which cell duplicates its genome. Genome means the basic set of chromosome. It is get duplicated, synthesizes the other constituent of cell and it divides into daughter cell. It is called as the cell cycle. So it is the definition of cell cycle. Eukaryotic cell represent the cell cycle in well elaborated manner. Howard and Pele, it is name of scientists Howard and Pele, in 1953 divided cell cycle as in these two categories, that is interphase and mitosis. he divide the interphase into G1 phase, S and G2 phase. And second part is the mitosis, that is multiplication phase. The human cells divide once in approximately 24 hours. Human cell takes the 24 hours for a complete cell cycle, while the yeast cell take only 90 minutes for a cell cycle. This is period of completion of cell cycle. This duration of cell cycle, it vary from cell to cell, type of cell or type of organism. So these two categories, that is interphase and mitosis, means cell cycle. We are starting from the interphase. The interphase also called the preparatory phase, or it is also called the intermitosis. The interphase, it is the stage between two successive M phases or mitosis. So it is called the intermitosis also. It is the longest phase of cell cycle, which lasts for more than 95% of the total duration of cell cycle. So it is a major part of cell cycle, that is interphase, during which the cell is highly active. Actually, it is metabolically active and prepare itself for cell division. It is also called the resting phase, although it is metabolically highly active. As the visible changes are not there, visible changes are seen in the M phase or mitosis. So the interphase is called the resting phase, although it is metabolically very highly active. The interphase is divisible into the G1 phase, S phase, G2 phase. During the interphase, the cell grows, accumulate nutrient needed for the mitosis, preparing it for the cell division and synthesis of DNA, RNA and most of the proteins. The interphase includes, you can see the figure, G1, S and G2 phase. Then the remaining part is the mitosis, including cytokinesis. Cell may enter into new cell cycle or may goes to the resting phase, that is G0 phase. We are going to see the G0 after G1 phase as separate part. In G1 phase, in this cycle, it is given as cellular content excluding the chromosome are duplicated. Means other than chromosome, all the content of the cell, remaining other content of the cell get duplicated. It is G1 phase in simple words. It starts with the completion of M phase of the previous cell cycle. Hence it is also called the post-mitotic phase. Although the G1 as well as G0 both are called the post-mitotic phases. If it goes to the G0 phase, it is not in the new cell cycle, it is also G0 phase, also called the post-mitotic, and G1 is also referred as the post-mitotic phase. The G1 phase occupies about 30 to 50% time of the complete cell cycle. 30 to 50% time of complete cell cycle, it is G1 phase. The cells are metabolically active and continuously grows. The G1 phase is marked by the process of transcription of rRNA, tRNA and mRNA. So it is characteristic feature of G1 phase. There is transcription of rRNA, ribosomal RNA, transfer RNA and messenger RNA, and synthesis of various types of proteins, enzymes and nitrogenous bases that are necessary for the synthesis of DNA. In G1 phase, protein synthesized during G1 phase includes regulatory protein. This regulatory proteins which control various events of mitosis, enzyme like DNA polymerase required for the DNA synthesis, tubulin and other mitotic apparatus protein. So in G1 phase, regulatory proteins are formed, including the DNA polymerase enzyme, tubulin protein and mitotic apparatus protein. During this phase, chromosomes are in the form of slender fine thread forming the intervening chromatin structures. The DNA synthesis does not occur during this stage. Hence it is called as the first gap phase. There is no synthesis of DNA. So it is called as the first, ये पहला gap phase होता है जिसमें DNA synthesis नहीं होता है, और DNA other than DNA, all other organs, DNA छोड़ के जो भी other organelles है cell में, they get duplicated in the G1 phase. So after that we are going to see the G0 phase. G0 phase, the word post-mitotic is sometimes used to refer both the quiescent and senescent cell. So it is the quiescent means G0, senescent means G1 phase. Some cells in the adult animal do not show division. For example, heart cell divide only occasionally, so they goes into the G0 phase as needed to replace cells that have been lost because injury or cell death. This non-proliferative, that is non-dividing cell, this cell does not divide. जो cell divide नहीं होते, that cell G0 phase में जाते हैं. Enter and inactive stage called as the quiescent stage or G0 stage of the cell cycle. These cells may remain quiescent for long period, possibly indefinite period. This is very common for cells that are fully differentiated. Some cells enter in G0 phase semi-permanently, not permanently in G0 phase. For example, some cells of the liver, some cells of the kidney cells, they may enter into the G0 phase. You can see here the G0 phase. Then G1 phase, we have already seen it. The cell from G1 phase, after duplicating the all other organelle other than the DNA, enters into the S phase. So we are going to see the S phase now. The S phase is also called the synthesis phase. The DNA synthesis or DNA replication, same thing, DNA duplication or replication takes place during this phase. So it is called the synthesis phase. S phase occupies roughly 35 to 45% of total cell cycle timing. The amount of DNA is doubled. Means if 46 chromosome in the human cell, that become 92 chromosome. So the number of chromosome double or DNA is double. If the initial amount of DNA it is 2C, then it increases to 4C. Means diploid cell become tetraploid. S phase occupies roughly 35 to 45. This thing is already repeated. Histone proteins are also synthesized during this phase. Histones, DNA synthesis, as well as there is synthesis of histone. So in the S phase, DNA are replicated or duplicated. In addition to this, there is formation of histone protein. Then we are moving towards the G2 phase. G2 phase, it is second gap phase. In the figure, it is written as the cell double check the duplicate chromosome for error, making any needed repair. The synthesis of DNA takes place in the S phase. They are checked here. If there is any error in the sequence of nucleotide, it is get checked and it is get repaired. It is the second gap phase and growth continues. So cell continue to grow and the protein are synthesized in preparation for mitosis. During G2 phase, synthesis of rRNA, mRNA and nuclear RNA takes place. So it is one characteristic feature of G2, synthesis of rRNA, mRNA and nuclear RNA takes place. It occupies about 10 to 20% time of total cell cycle. By the end of this stage, cell content increased twice. This stage has great significance because check duplication of the chromosome for the error, making any needed error, or it is get repaired in the G2 phase. Then we are moving toward the M phase, that is mitosis. So mitosis, it is also referred to the period of division, or it is called the M phase. Mitosis meaning threads. So thread like appearance of chromosome are there, they are clearly visible, so it is called the mitosis, means threads. M phase involves, or mitosis can be divisible into these two large categories, that is karyokinesis and cytokinesis. Here karyon means nucleus, so division of nucleus means karyokinesis. Cytoplasm, division of cytoplasm is cytokinesis. So karyokinesis plus cytokinesis equal to mitosis. So karyokinesis is the division of nucleus into two daughter nuclei, whereas the cytokinesis is the division of cytoplasm resulting in two daughter cells. So first we are going to see the karyokinesis. There are four subtypes, that is prophase sub-stages: prophase, metaphase, anaphase and telophase. So we are going to see one by one. That is the first one is the prophase. In the prophase stage, the nuclear membrane starts disappearing. First thing, nuclear membrane starts to disappear and the nucleus with thin coiled thread-like chromosome is seen. These chromosomes are chromatin, referred as the chromatin. They are up to thin and they starting to become thick and short and coiled. So prophase marked by the following character: chromosome material condensed to form compact mitotic chromosome. Chromosomes are seen to be composed of two chromatid as they are replicated. So each chromosome looks as a two chromatid attached together at the centromere. Centromere of these two chromosomes get attached to each other. So they look as an X or cross due to presence of sister chromatid together. Their centromere get attached as they are replicated. Then second thing, centrosome which had undergone duplication during the interphase. Centrosome जो interphase में ही तैयार duplicated हो जाता है, begins to move towards opposite pole. वो एक दूसरे से goes away from each other toward the opposite pole. So they reaches to the opposite pole. Each centrosome radiates out microtubules called aster. The two aster together with the spindle fiber, they form the mitotic apparatus cells. Then chromosomes are arranged at the periphery of the nucleus. Mitotic apparatus is almost completely formed in the prophase. Then second one is the metaphase. Then we are going to move toward the metaphase now. Metaphase can be divisible into pro-metaphase and metaphase itself. Pro means the beginning of metaphase or early metaphase, and it is the complete metaphase. In the metaphase, chromosomes are completely condensed so that they appear very short. All the chromosomes lie at the equatorial plane of the cell. This is called as the metaphase plate. The equatorial plane, it is called as the metaphase plate. Mitotic spindle is fully formed. Metaphase is the stage at which morphology of chromosome is most easily studied. So all the study of chromosome, it is done in the metaphase stage due to chromosomes are bold, short and clearly visible and thickest in the metaphase. They found at the metaphasic plate. Small disc-shaped structure at the surface of the centromere is called the kinetochore. It is the structure served as for the attachment of spindle fiber. This egg-shaped sister chromatid, they join together by the two centromeres. On outer side of this chromatid, rounded structures are formed. They are called the kinetochore. It is the attachment of spindle fiber for the separation in the next phase, that is the anaphase. So we are going to see the anaphase now. The anaphase, the chromatids of each chromosome separate and form two chromosomes called as the daughter chromosomes. The formed chromosomes are pulled away in opposite direction by spindle apparatus. This kinetochore, to the kinetochore, the spindle fibers are attached. This kinetochore gets shortened and chromosomes get pulled toward the opposite pole. So they become in this way inverted V shape, or they become V shape in opposite pole. They get pulled toward the opposite pole. The key events of the anaphase: centromere split and attached to the kinetochore, chromatid move toward the opposite pole. So then last one is the telophase. Then we are moving toward the telophase. Telophase is the final stage of the karyokinesis. So telophase, it is the final stage of karyokinesis. The chromosome at the poles began to uncoil, lengthen and lose their individuality. These words are very important. Chromosome at the opposite pole began to uncoil. They become relaxed. They become short in the metaphase. They become uncoiled, short and become lengthened, and they relax and they lose their individuality. So they get mixed, in other words. The nucleus begins to reappear. Nucleolus तैयार शुरू हो जाता है, begins to reappear. Nucleolus inside the nucleus. The nuclear membrane begin to appear around the chromosome. So दो nuclear membrane तैयार होते हैं एक सेल में. At each pole around the group of chromosome, nuclear membrane starts to form. This is the nucleolus inside this. So two nuclei are formed. Spindle fiber break down and get absorbed in the cytoplasm. Thus two daughter nuclei are formed in the cell. Key events of the telophase: chromosome cluster at opposite spindle poles and their identity is lost. The nuclear envelope develops around the chromosome cluster at each pole, forming two daughter nuclei. Nucleolus, Golgi complex and ER reform. In addition to the nucleolus, outside the nucleus, Golgi complex and ER also form in the cell. So this is the telophase. Then cytokinesis is the part after karyokinesis. So in the karyokinesis we have studied the prophase, metaphase, anaphase and telophase. The karyokinesis, the division of cytoplasm into daughter cell, is called the cytokinesis. Kinesis, break of the cytoplasm or division of cytoplasm, is called the cytokinesis. It starts with the constriction, which gradually deepens and ultimately join in the center, dividing it into two daughter cells. It is characteristic feature of animal cell. You can see in the figure, this is the constriction which goes deepen and deepen. Finally they get separate into two cells. This constriction appear at the middle, but this thing is found in the animal cell. However, in the plant cell, they are covered by a relatively non-flexible cell wall. A different thing is formed between the two cells, that is cell wall. The formation of new cell wall begins with the formation of simple precursors called the cell plate that represent the middle lamella between the walls of the two adjacent cells. At the time of cytoplasmic division, organelles like mitochondria and plastids, they get distributed between the two daughter cells.
So this is about the cell division and mitosis. So I think you got everything, each and every point from the cell division. All the phases, S phase, G1 phase, G1 S and G2 phases, as well as prophase, metaphase, anaphase, telophase from the mitosis. The next part we are going to see in the next lecture, that is the meiosis, distinguishing features between the mitosis and meiosis also. Meiosis, it is divided into two parts, meiosis first and second. So we are going to see the meiosis first, how we enter into meiosis second, and what are the stages in the meiosis first as well as meiosis second and cytokinesis. So see you again. So thank you very much. That's about this lecture.